Students, good morning to everybody. I welcome you all, both our students as well as from outside. Now you are also our students because you are already admitted in the school Alpha. As per the director's instruction, we are just actually taking classes to you through online because it's not possible during this lockdown period. So I want to communicate, I want to interspace through these online classes only. Now we are talking about mainly the science subjects. The science subjects include the different fields, either mathematics or physics or chemistry or biology. I am a biology teacher, teaching biology for a long time in the school. Many of you know and many of you do not know me. Now, we have to know something about biology. I am not taking what we have the class. We are introducing just actually the concept related to some of the what is called the principles, what we are actually just facing in our today life. Now, you know that one, biology, the meaning for that one. The word biology was given by Lamarck. Many of you do not know, the word was given by or coined by Lamarck. An evolutionist, a French evolutionist, just actually made the concept of evolution, wrote a book also. So now in foreign countries, the subject has been renamed as life sciences. So the study of life, either the animal life or the plant life, including the human beings. Now we have a number of fields in biology: anatomy, physiology, cytology, histology, and so on. But we have to concentrate more and more as we are entering into a new arena, that is 11th standard, and we are going to stay in that school for 2 years to acquire knowledge about the science, to acquire knowledge about biology, and leaving the school with more information, and also literally as well as academically. It will be helping you normally in future for a better future. I think so I can do well for you whatever just the concept we have in biology in subsequent classes. Now I have to tell some of the important actually the fields, the advanced fields. Now at present you have to know, in addition to the concepts of anatomy, then physiology, cytology, etc. Now the advanced technology in biology is biotechnology, one of the fields. We can get more information, we can get more knowledge, a new field. So it is nothing but you see that one, harvesting products useful to human beings from the microorganisms, reaping or harvesting, obtaining from microorganisms. And also the entire microorganism of the products, they are useful to us, that field biotechnology. Then we are changing the genetic nature of the organism by introducing or inserting a new gene so that it can produce a number of products useful to humans, either medically or for various purposes. For example, by introducing the gene for the production of insulin hormone, one can able to synthesize large molecules of insulin because body can synthesize only a few molecules of insulin in the case of diabetic patients. For a substitute, we have to synthesize insulin hormones through a method what's called genetic engineering. Such a hormone which is being synthesized through genetic engineering is called humalin. The hormone which is synthesized through genetic engineering. So in genetic engineering we are constructing a new organism by incorporating a gene into that organism. What we can say the animals or the plants, transgenic organisms. Organisms with its own what is called the genetic material along with something material, the genetic material from other organisms. For example, the golden rice. The golden rice, you see that one, it's not made of gold. It is being synthesized through genetic engineering. You know, the rice or paddy has no ability of synthesizing vitamin A. So that scientists incorporated the gene into the plant rice paddy and synthesize and making them to synthesize what we have, that is vitamin A. Such an organism, such a paddy which can synthesize vitamin A because of genetic engineering method is called a golden rice. Now again, we have normally writing everything and storing the informations, whatever the scientists have developed, whatever the scientists have invented, whatever the scientists have discovered, only in notes. In due course, the materials, the book materials, or the materials in the notebook may disappear. In such cases, we have to store information in the form of a program in a computer. So, we have the biotechnology plus information technology together form what is called bioinformatics. So it is nothing but actually synchronized or integrated study of BT plus information technology. BT, biotechnology and then information technology. So this is nothing but the creation of electronic databases for biological information, either about the nucleic acids, or about what we call the gene sequences, or we call the amino acid sequences in a protein. That field is called bioinformatics. Okay, creation and maintenance of electronic databases. That is integrated study of biotechnology and information technology. Even we have also many informations have been made by the scientists about, for example, the color perception. Is only human beings or the ability of just perceiving the colors, or the animals too? We have to find out. So, when compared to human beings, there are certain animals can perceive the colors more effectively. So you have to find out the answer for that one. Which animal can perceive the colors more effectively? It may be one of the assignments for you. Remember that one, which animal can perceive more colors. Now we are also getting more and more information through what is called the computer, internet, there is a interfaces. Even also there is a what is called conversation about the brain of Einstein. So scientists have normally made, the doctors have made research studies about the brain of Einstein. So he died, you know that one, Albert Einstein, in 1955. So that brain, his idea was his body to be cremated and his body would not be given to anybody. But accidentally one doctor just actually sealed his brain, captured his brain without known to anybody. And later, after nearly 50 years, the brain was supposed to, for experimental analysis by the scientists, they found some differences. Now find out the differences between our brain with that of Einstein's brain. In what way Einstein's brain is superior than humans? These are all the new concepts you have to actually develop. So about the coloration, about bioinformatics, about the brain of Einstein, etc. Like that we can go on just actually searching so many. Even nowadays, there is also a method, what is called gene therapy. Gene therapy. What is therapy? Therapy means treatment. We know physiotherapy, chemotherapy, magnetotherapy, theotherapy. What is theotherapy? Therapy means treatment. So we have what is theotherapy. So when you have fever, what would happen? Your parents are taking to you either to what we have just actually the mass or to the temple Now the one who is there just actually sprinkling water or sprinkling what we have even just actually ash, etc. So treatment through religious methods. You know well, treatment through religious methods. That is called theotherapy. So likewise, gene therapy, the new advanced method, but it's a costless one. What do you mean by gene therapy? So replacement of defective genes with the corrective genes. If a particular gene is defective, say an example, the gene producing the insulin hormone is defective, so that the person develops diabetes. In such cases, the defective gene has been replaced by what is called the normal gene. So that is treatment of genetic disorders by correcting or by replacing what is called just defective genes with the normal genes. That's a new method. So likewise, we have a number of methods adapted. So we are all interested to go for medicine, to become a doctor. But more than a doctor, we have a number of paramedical courses. For example, nursing, radiology. It's the best one nowadays, you know that one. Using the different techniques of MRI scan, etc. Radiology. Even MLT group, you know that one. Even just we see food processing technology. This is also another method. Just actually we can earn more and more also. That is what is called the job wise we can have. Now the biotechnology can go for research field, and also we have been taken by the different food processing industries. For example, the Cadbury, the Nestle, the Parley. Okay, these are all the food processing industries. They need badly a person with biotechnology degree. So, not always thinking about what we have the medicine. Other than medicine, research field we have more and more. So I can gain more and more knowledge while studying such things. Okay, and also we have been salaried through this job. What we have in the case of Parley, Nestle, etc. Extensive research work has also been made just about the behavior of animals. The study of behavior of animals is called ethology. The study of behavior of animals is called ethology. Say an example, number one, do just animals dream? Do animals dream? Is it possible for animals just like human beings to dream during sleep? Or we can say the dog, just regarding the dog can just feel the magnetic field. This is a peculiar behavior for the dog. That's why they can feel better. Either tsunami is coming, or any other earthquakes, or any other calamities or catastrophes are coming to what is called the land. They can feel better. They can also find out easily even before human beings can find out. So do animals dream, or dog can feel the magnetic field? These were the research works conducted by the scientists. They also got information about these things. Likewise, we see the plants also conduct the electric field through their root system. This is another thesis has been made by the scientists recently. So these are some of the new area. You have to look into the internet and find out the answer. So I am not giving the answer for that one. You have to find out whether the animals are dreaming during their sleep or not, or either the dog can feel the magnetic field or not. Okay, now with this introductory part, I want to give an important just part one must know in your life, including your parents also. That is what we have the blood group analysis. Each and everyone must know just your blood group. How to find out the blood group? How far the blood groups have been classified? And how can you know your blood group? What is called positive blood group, negative blood group? Okay, I want to take this one, blood group analysis. So now we have to go for somehow related to the subject, blood group analysis. In the blood group analysis, I would like to take the first one, ABO blood system. Now we have to study the blood group analysis. Everyone must know it, that to the ABO blood system. Because many students normally do not know their own blood groups. Why I am saying that one, the last day also one of the students that is frightened about what is called the syringe or injection. Till today, that person do not know just her or his blood group. So it is very important because blood transfusion is a must nowadays, you know that one, because of trauma. Trauma means injuries due to impact of force, due to what we call the accident, very common nowadays. So one must know the blood system, that is his or her blood group. Now the ABO blood system was first reported by just actually Carl Landsteiner in 1900. He is called father of hematology. Hematology means study of blood, not blood group, study of blood. So first discovered, that is by Carl Landsteiner in 1900. You know there are 4 different types of blood groups. We'll see the subgroups also later. A, B, AB and O. Out of the 4 blood groups, he discovered only 3 blood groups, A, B and O only. The rarest blood group, that is one of the questions also, the rarest blood group, that is AB. It's the rarest blood group, least percentage in the population. Was discovered by 2 of his students. The students who were working under him, they just actually observed what is called the 4th type of blood group, the rarest blood group, AB. So, Warney Casserly and what we have Sterling. These were the 2 students of Carl Landsteiner observed the rarest blood group AB in 1902, 2 years later. That's about something the historical aspect. Now on what basis the blood groups have been classified? Carl Landsteiner observed certain substances present on the surface of RBC membrane. He called those substances as antigen. We call those substances as antigen. What is an antigen? Why is it called so? Now let's see now. What is the general meaning of antigen? So the antigen, normally you see that one is called so because it is the antibody generator. So it is an antibody generator. What's the meaning? When a foreign substance enters into the body, either a food or a microorganisms, their products, even the pollen grains, they are highly toxic, you know, the pollen grains. Then the pollutants, any other chemicals, even the sweet coating of a tablet or the capsule of a tablet. And that may cause the stimulation of certain protective proteinaceous substances in the plasma. Such proteinaceous substances formed in the plasma, which are protective in nature, giving defense to our body, are called antibodies. I'll come back to this one later. So when antigen enters in the body, it stimulates the production of antibody. Hence the name antigen is given because it generates antibody against it. So we have antibodies generally. Now generally the antigen is called a foreign substance. What's the meaning of foreign? Any substance which will not be accepted by the body. Some persons can accept the sweet coating tablet. Some persons cannot accept sweet coating tablet because it causes changes in the body. Now here the sweet coating of the tablet is an antigen because it's not accepted by certain persons, not by all. So antigen is a foreign substance. What is a foreign substance? Anything which is not accepted by the body. Some cases you know, even eating bhaji is also not accepted by some persons. It stimulates what is called the sneezing and coughing. In such instance, it is called as a foreign substance. Okay, that is not accepted by the body. So which includes either the microorganisms, the toxic products, the pollen grains, the pollutants. or the chemicals, etc. So anything which is not accepted by the body. This is a general statement. But we are talking about what's called the blood group antigen. Where is it located? The blood group antigen is located on the surface of the plasma membrane of RBC. Now this is RBC, this is the antigen. Blood group antigen is located on the surface of RBC. For example, see, it is present on the surface of RBC and it's normally made of organic substances, sucrose, D-galactose, N-acetyl galactosamine, amino acids, etc. It is a combination of many organic substances. This is not necessary now, simply organic substances collection. So, when a person having blood group A donates blood to B, what is happening is not accepted. When this blood group containing antigens stimulate the production of antibodies in the blood of B. I'll come back to this one. What is an antibody? So once antibodies are produced here, they fight against antigen, leading to the clumping of blood cells, resulting in the death of the individual. So this is what we can say agglutination. Clumping, sticking together of blood cells like this. All the cells of RBCs are stuck together, so rendering just actually unable to. When they are individual, they can. Once they are all aggregated, this is called agglutination, sticking together. This is because of the introduction of an antigen opposite to the blood group B. Okay, this is one. So what is an antigen? It's a foreign substance. What is an antibody now? So an antibody is a protective proteinaceous substance produced by the stimulation of antigen. That's why it's called as antigen is nothing but antibody generator. Only when antigen enters the body, it stimulates the production of antibodies. Now what are antibodies? Antibodies are protective proteinaceous substances because they are made of a protein immunoglobulin. When are they produced? They are produced by the stimulation of an antigen. Produced by the stimulation of an antigen that is actually in the plasma. While antigens are present on the surface of RBC, antibodies are present in the plasma. Just imagine that one, understand that one, recall that one, retain that one in your memory. Antigen on the surface of RBC, antibodies in the plasma. The universal name for antibodies, agglutinins. So the antigens are called agglutinogens, the antibodies are called agglutinins because they cause the agglutination, the sticking together, clumping together of RBCs. Hence the name agglutinins. So by the stimulation of an antigen, now I will represent that one also by means of a reaction. So when reaction occurs between antigen present in RBC against opposite antibody in plasma, we have the agglutination or clumping of blood cells, nothing but sticking together. See, all the RBCs are stuck together to form a mass. What is the importance of this antigen-antibody reaction? The antigen-antibody interactions or the reactions help to determine the blood groups in blood transfusion. That's the basis. One can find out the nature of the blood group, what type of blood group that person has only by means of the interactions or the reactions between these two. Now, what is the basis for classification of blood groups? What is the basis for classification of blood groups? On the basis of presence or absence of antigen or agglutinogen on the surface of RBC, we can classify the blood groups into four different types: A blood group, B blood group, AB and O. We can see the subgroups also later. Now, here is that one. Landsteiner observed the presence of two different types of antigens. Any one of them or both are present on the surface of RBC or no antigens. The antigens he observed are antigen A or antigen B. So an individual may have either A antigen or B antigen or both antigens or no antigens. A antigen or B antigen, both antigens or no antigens. For example, A blood group, the persons have A antigen in the RBC. B blood group persons, they have B antigen on the surface of RBC. AB blood group, both A and B antigen. And O blood group, no antigen. So that's about what we call the classification. I'll show the illustration now. Through the diagram you can understand how far the blood transfusion is possible. In addition to the four common types of blood groups, there is a rare blood group called Bombay blood group, seen in India, Pakistan, Bangladesh and also in Iran. The frequency is 1 in 4 million people. And find out what is actually Bombay blood group on your own. Okay, now just I want to show the illustration how far the blood groups are classified. I am taking 4 samples of the blood groups A, B, AB and O. Now let's take what does the A blood group contain. I mentioned already the classification is purely based on the presence and absence of antigen. So A blood group persons have A antigen in their RBC. Now this is the RBC I mentioned about, this is the plasma. The B blood group persons have B antigen in their RBC. This is the RBC, have B antigen. Then AB blood group persons have both the antigens not in the same RBC. Suppose if you have 100 RBCs, 50 RBCs have A antigen and 50 RBCs have B antigen. So I am taking both A antigens and B. So O refers to 0, no antigens. That's why the blood group is called as O blood group. Suppose, let us assume this person is the recipient. The one who receives blood is called recipient. Let's see what happens when transfusion occurs with the different blood groups or the same blood group. So I am taking this individual as that is what we call recipient, the receiver. All the blood groups I am taking just as a receiver. That's all we can say, the technical word recipient, the receiver. I am using the word technical word, recipient. So these are all the recipients, receivers. I am taking here donor, the one who donates the blood. Okay. So I want to donate blood, A blood to A blood group persons. Let's take this one. So the donor, first I am taking this A blood group. A blood group is donated. Now the A blood group is accepted. No change occurs in the recipient body. Now I am introducing what is called B blood. The B blood cannot be accepted because when B antigen enters here, it stimulates the production of antibodies to fight against antigen B, hence called anti-B antibody. This is called anti-B. Anti-B. I'm taking. So, introduction of antigen B stimulate the formation of antibody B as it works against the B antigen. That's why the clumping or agglutination occurs. Suppose I am introducing AB blood group to this person, transfusion. It is also, so B blood group will not be accepted by what is called A. So I am just putting a cross marks denoting it is not being accepted. So this is an indication. I am putting a cross mark, not accepted. Similarly, when AB is introduced, it is also not accepted because 50% of the blood cells contain B antigen. It stimulates the production of what is called, that is, antibody B. So, the A blood group persons have A antigen in the RBC, anti-B antibody, that's the opposite antibody in the plasma. That is the nature of A blood group. Now let's go to the next one. Okay, here I am just giving what is called the O. Now the O blood can be accepted because in O there are no antigens. I mentioned already no antigens. So one of the principle use blood transfusion. The donor should not contain specific antigen against the antibodies of the recipient body. I will write down later. So the donor should not contain antigen against the antibodies of the recipient body. So here there is no antigen to stimulate either A or B antibodies. No, it can be accepted. So A blood group persons can receive A and O. I'll take the next one. So here I transfuse what is called A blood group. I am saying that one is not accepted. What is the reason for that one? So the plasma develops anti-A antibodies. The plasma develops anti-A antibodies. The one which is working against antigen A. So we have, actually the B blood group persons have B antigen in RBC and anti-A antibodies in plasma. Suppose when B is transfused, it is accepted. When AB is transfused, as a rule you know that one it contains what is called the antigen, that is A, so it stimulates the production of anti-A antibodies, so it is not being accepted. I put a cross mark indicating that it will not be accepted. Then what about O? When O is given, that will be accepted. So B blood group persons can receive, that is B, their own blood group and also the O blood group. Now coming to the AB blood group. So when A is given, I am giving A, that is accepted. When B is given, that is accepted. When AB is given, that is also accepted. When O is given, that is also accepted. What's the meaning for that one? So AB blood group persons have both A and B antigens, but there are no antibodies in the plasma, either anti-A or anti-B to fight against antigen A, antigen B. That's why AB blood group persons can receive blood from all the 4 different types. Hence this AB blood group is called universal recipient. Universal recipient. That too, that is what is called AB positive, not AB negative. I will tell you more about the information later. So it is called universal recipient, particularly AB positive, not AB negative. So this is one. What about O blood group? When A is given, so when A is given, when B is given, when AB is given, all these what we have the blood groups cannot be accepted by O. The reason for that one, O blood group do have no antigens having both the antibodies A and B. So we have in the plasma anti-A and anti-B antibodies. Hence reaction occurs between antigen and antibody. So antigen reacts with anti-A, B antigen reacts with anti-B. What about O? O can receive its own blood group, no problem. O can receive its own blood group because there is no antigen in the case of O blood. Now from this one you can see what you know about A blood group. Having antigen A RBC, antibody B in plasma. Then B blood group having anti-A, just antibody in plasma, then B antigen RBC. Then AB, they have both antigens A and B, but there are no antibodies. Then O blood group, they have both the antibodies in the plasma, but there are no antigens. So the difference between that is AB and O, it contains both the antigens, no antibodies. It contains both the antibodies and no antigens. Now here in all the blood groups can receive what is called the O. Hence O blood group is called universal donor. Universal donor. That too what we have, there is O negative. O negative is universal donor. So that's about the classification. You can represent the same one also, that is in the form of, that is what we have the tabular column. Now the most abundant blood group is O positive. The most abundant blood group is O positive. The least abundant blood group is AB negative. The least abundant blood group is AB negative. Normally 80% of the individuals are positive. Either A positive or B positive, or AB positive or O positive. Then we have also A negative, B negative, AB negative and O negative. These are the 8 types in addition to A1 also. So I will come to this one, the positivity and negativity. But here the most abundant blood group is O positive. The least abundant blood group is AB negative. The universal donor is O negative. The universal recipient is AB positive. Remember this one. And we have also the AB blood group is an example for codominance. We'll discuss under the genetics. Now I will represent the same one in the form of tabular column. You can just copy down and remember it. Based on the illustrations what I taught, I have given everything in the form of tabular column. Say blood group A having antigen A, having antibody B. It can be donated to A and AB. It can receive blood from A and O. Then B having antigen B, anti-A antibody in the plasma, that is B arm can donate to B and AB and can receive from B and O. That's a universal donor. Now AB having both antigens A and B, but there are no antibodies. That's the reason why it can just receive blood from all. Then just can donate only to AB, its own blood group, but can receive from all A, B, AB and O, hence called universal recipient. Then O blood group, no antigens, that's the reason why it can be given to all. Then both anti-A and anti-B, just what we have the antibodies, it can be given to all A, B, AB and O, hence called universal donor. That's why I mentioned about O negative. Then here this is what's called AB positive. So AB positive, this is what we have O negative. Even under O blood group, O negative is a universal donor. It can receive blood from just actually only O. So that's about the blood group analysis. I want to give one statement. So the blood group transfusion depends on or based on the following fact. What is that fact? The donor should not contain antigen against the specific antibody of the recipient serum. That is a principle used in blood transfusion. Okay, the donor should not contain antigen against specific antibody of the recipient serum. For example, A blood group persons actually they have B antibodies, that's why B cannot be given to A. So they should not have opposite antibodies in the plasma. So this is about what we have the blood group analysis. We have more information later about the RH blood group. Okay, so I will give this lecture through what we have the video. Along with that video, I will send another video of 4 minutes. And also we have the question. We have the one-word answers and also 2 marks question. Answer it as per the rule, upload it. Okay, that is want to just understand for others. Normal blood group and the basis of classify பண்றாங்கன்னா antigen இருக்கா இல்லையா, என்ன antigen இருக்கோ அதை பேஸ் பண்ணி blood group classify பண்றாங்க. A antigen இருந்துச்சுன்னா A blood group, B antigen இருந்துச்சுன்னா B blood group, ரெண்டுமே இருந்துச்சுன்னா AB, antigen இல்லைன்னா O blood group. ஒரு blood groupல பார்த்தீங்கன்னா ஒரு சாம்பிள் எடுத்தீங்கன்னா blood group A-வா இருந்துச்சுன்னா RBCல antigen A இருக்கும், படத்தில் பார்த்தீங்கன்னா தெரியும். ஆனால் பிளாஸ்மால anti B, அதனால B blood group வந்து blood group B blood group வந்து Aக்கு கொடுக்க முடியாது. அதே மாதிரி B blood group பார்த்தீங்கன்னா RBCல B antigen இருக்கும், ஆனால் antigen A-வை எதிர்க்கக்கூடிய anti-A antibody பிளாஸ்மால இருக்கும். ஸோ அதனால A வந்து Bக்கு கொடுக்க முடியாது. இப்போ AB வந்து நார்மலாக ஜஸ்ட் ABக்கு மட்டும்தான் கொடுக்க முடியும். ஏன்னா அதில் 2 ஆன்டிஜனுமே இருக்கு. அதே நேரத்தில் ஏபிக்கு வந்து எல்லாமே கொடுக்கலாம். ஏன்னா ஏபியில் வந்து ஆன்டிஜன் மட்டும்தான் இருக்கு, ஆன்டிபாடிஸ் கிடையாது. அதனால ஏபி எல்லாத்தையும் பிளட் வாங்கிக்கும். ஆனால் ஏபி கொடுக்கறது ஏபிக்கு மட்டும்தான். ஏன்னா ஏபியில் ஆன்டிஜன் இருக்கு. நான் தான் பிரின்சிபல் சொன்னேன், பிளட் கொடுக்குறவங்களுக்கு அந்த ஆன்டிஜன் இருக்கக்கூடாது, ஆன்டிபாடியை எதிர்க்கக்கூடிய ஆன்டிஜன் இருக்கக்கூடாது. இப்போ ஓ பிளட்ல பார்த்தீங்கன்னா ஆன்டிஜனே கிடையாது, அதனால அது 0 O. போத் ஆன்டிபி ஆன்டிபாடிஸ் இருக்கும். அதனால பிளட் குரூப் ஓக்கு யாருமே பிளட் கொடுக்க முடியாது எக்ஸப்ட் ஓ. ஏன்னா வந்து அந்த ஆன்டிபாடிஸ் இருக்கு. ஏ கொடுத்தாலும் சரி, பி கொடுத்தாலும் சரி, ஏபி கொடுத்தாலும் சரி, அவங்க ரியாக்ட் பண்ணுவாங்க ஆன்டிபாடி கூட. அதனால ஓ வந்து ஓ இது மட்டும்தான் ரிசீவ் பண்ணும், ஏன்னா ஓல ஆன்டிஜன் கிடையாது. ஸோ இதுதான் பிரின்சிபிள். ஓ ஆன்டிஜன் வந்து இருந்துச்சுன்னா அது கொடுக்கக்கூடாது, ஆப்போசிட்ல இருக்கும். திஸ் இஸ் த பிரின்சிபிள். உங்களுக்கு ஏதாவது டவுட் இருந்தால் கேட்கலாம், அப்புறம் ஜஸ்ட் ஐ வில் எக்ஸ்பிளைன். இது பேசிக் பிரின்சிபிள் நான் சொன்னேன், யார் கண்டுபிடிச்சா என்ன. இப்போ பாம்பே பிளட் குரூப்னா என்ன கண்டுபிடிங்க. ஓகே, these are all some of the actually points related to blood group. Then have to go further in the next class regarding the RH fact. Okay, so the AB positive is a universal recipient and O negative is a universal donor. ஏன் O நெகட்டிவ்னா ஓல ஏ ஆன்டிஜன் கிடையாது, பி ஆன்டிஜன் கிடையாது, இன்னொரு ஆன்டிஜன் RH ஆன்டிஜன் சொல்லுவாங்க, அதுவும் கிடையாது. அதனால O கொடுக்கலாம். ஏபி பாசிட்டிவ்ல பார்த்தீங்கன்னா ஏ ஆன்டிஜன் இருக்கும், பி ஆன்டிஜன் இருக்கும், இன்னொரு ஆன்டிஜன் RH ஆன்டிஜன் இருக்கும், அதுக்கு பேர் தான் பாசிட்டிவ் பேர், அல்லது டி ஆன்டிஜன் சொல்லுவாங்க. ஆனால் எந்த ஆன்டிபாடியுமே கிடையாது. அதனால ஏபி பிளட் குரூப் எல்லாத்தையும் ஆயிடும். So this is a nutshell. You can understand, I think so. We will meet once again in the next class. Okay, thank you.